Okay, so anaerobic bacteria, aerobic bacteria, anaerobic bacteria, anaerobic bacteria, they are further classified into anaerobic bacteria, they are further classified into facultative anaerobes and obligate anaerobes. Facultative anaerobes mean that they can survive both in aerobic and anaerobic conditions. Like these bacteria, whenever there is a shortage of oxygen, they can switch their metabolism from aerobic respiration to anaerobic fermentation or anaerobic respiration. So they can even survive in the presence of oxygen, they can even survive at the absence of oxygen. So these bacteria, facultative anaerobes, most common example for this facultative anaerobe is E. coli. So Escherichia coli bacteria, it can survive uh, in uh, uh, in the presence of oxygen as well as in the absence of oxygen. So once you know that the respiration, what is respiration? Conservation of oxygen. So if the bacteria there is no oxygen, then they can ferment some other pathways to produce ATPs to generate energy. So that's how this facultative anaerobic bacteria, they survive in the absence of oxygen. The next group we have the obligate anaerobes. So obligate anaerobes, obligate anaerobes, these bacteria, the most common example for these bacteria is Clostridium tetani, C. tetani, Clostridium tetani. So Clostridium tetani is the example of the bacteria which survive, which is totally anaerobe, obligate anaerobe. It means they cannot grow in the presence of oxygen. This bacteria cannot grow in the presence of oxygen. Why this bacteria cannot grow in the presence of oxygen? Because this Clostridium tetani does not have the superoxide dismutase and catalase enzyme. So this bacteria does not have, this bacteria does not have any enzyme. Which enzyme? I explain you about catalase and dismutase enzyme that stabilize the oxygen and oxygen related metabolite. So this bacteria do not have these enzymes, so that's why this bacteria cannot survive in the condition in which there is an oxygen. So the next we have microaerophilic bacteria. Microaerophilic bacteria are those bacteria which can survive in a very low concentration of oxygen. They does not require a high amount of oxygen, and these bacteria, they can be killed if high oxygen concentration is available. If they are exposed to the high concentration of oxygen, they will be killed, but they can survive in a small or very you can say a very low concentration of oxygen. So other regulation, uh, like, so this is the oxygen requirement of the bacteria. So next we have a growth curve or the growth of bacteria. What is the growth of bacteria? How bacteria grow? I explain you about like at the beginning I explain you that the bacteria grow exponentially with the by binary fission. So one bacteria convert into 2, 2 into 4, 4 into 8, 16 and so on. So when bacteria replicate, there are four phases of bacteria. There are four phases of bacteria. This is known as growth curve. So what is growth curve? The growth curve is if a bacteria are inoculated into a liquid nutrient media, and the number of bacteria are counted at a frequent interval, then we can get these, then we can get this curve. I'm saying, for example, when we have a Petri plate or we have a media, we grow the bacteria in the lab, in the laboratory. So when we grow the bacteria in the laboratory with the presence of nutrition, and at different interval of time, 10 minutes, 20 minutes, 30 minutes, 1 hour, 2 hour, 5 hours, 10 hours, 20 hours. So with the passage of time, with the time, we take the bacterial sample and count the bacterial number of bacteria, and count the number of bacteria, like after 10 hours or maybe 1 hour, how many new bacterial cells are formed, how many, and after 10 minutes, how many bacteria they are formed, they are multiplied. So when I count this number of bacteria, so I can get this curve. So this curve has 4 phases. So bacterial growth, when bacteria is grown in a lab, or even if a bacteria is grown in our body, within our body, there are 4 phases. One is lag phase, second is log phase, third is stationary phase, and the fourth one is death phase. So there are 4 phases of the bacterial growth. This is known as the growth curve. So what happened in these 4 phases? During the lag phase, this is the first phase. In this phase, the bacteria, there is no growth of bacteria, but this phase, there is a vigorous metabolic activity occur. Like the bacteria prepare itself for replication. So before replication, bacteria need to acquire or need to reserve some metabolites for replication. For example, if you want to go for a picnic, so you need to make a plan, and you need to buy some things, some food for the picnic. The same, the bacteria, before replication, bacteria need to reserve some metabolites, some nutrients which are required for replication. So during the lag phase, there is no bacterial growth, but bacteria are reserving the nutrients, and there is a high metabolic activity in the cell, but there is no cell division. So this lag phase, it can last for few minutes to maybe many hours. It depends on the bacteria. Okay, it depends on the bacteria. The second phase is a exponential phase, and that is also known as log phase. Okay, this is also known as log phase. In this phase, the bacteria replicate rapidly. So there is a rapid replication. So here you can see in log phase, the number of bacteria, it increases. So during the log phase, the bacteria replicate. And we know that when the bacteria replicate and new bacteria are formed, so what will happen? There will be a shortage of food, shortage of nutrition. If you increase the population of the world, so what will be the like side effect or the major adverse event occur? That will be a shortage of food, shortage of nutrition. So that is why you can see in some countries, underdeveloped countries, the population is more, but the food resources are very less. So that is why the death rates in those countries are very high. So same in bacteria. In once the bacteria achieve the maximum growth, now there will be a shortage of nutrition. Because of shortage of nutrition, as well as the bacteria produce some metabolites which kills the surrounding bacteria. So after the log phase, the third phase will start, that is a stationary phase. This phase, this stationary phase, the rate of growth or a number of new cell, production of number of new cell is equal to number of deaths. In the stationary phase, the number of new cell formation will be equal to the number of deaths. So that is why for a certain period of time, there will be no net growth occur. No net growth occur, because the number of deaths will be equal to the number of new cell form. So the stationary form or the stationary curve will achieve. This is the third phase. The fourth phase is the death phase. In the death phase, it's the last phase in which the bacterial cell going to die. Why? Because of the lack of nutrition and metabolites, toxic metabolites, which are produced by the bacteria. Lack of lack of nutrition, production of the metabolites, secondary metabolites, which kills the surrounding bacteria. So at this phase, because the nutrition has been depleted, so no new cell will be formed, but the number of deaths will increase. So ultimately, the bacterial growth will become less and less. So this curve, this four phases curve, is known as the growth curve of a bacteria. Yes, class, understood?
Yes,
You
sir.
will study in community medicine uh, about this. Let's suppose we have 5 prisons. We have 5 prisoners, sorry, prisoners, and we have locked them in a closed jail or a prison. So 5 prisons, and the jail is the room size is very small, and there is only a single window in the room. So after 10 days, all people died due to which reason? We have a very small window here, and we have a very small room, and five prisoners, they are in that room. So after 10 days, all the 5 prisoners, they died. So what might be the reason?
It is just there.
Yeah, suffocation, hypoxia or suffocation, because there is no ventilation. So the same in bacteria, in the fourth phase, in the death phase, there is no nutrition, so bacteria will starting to die. So this is we call the growth phase. Next we have the fermentation process. It is the process through which a bacteria gets the energy. You know, fermentation process is what? So fermentation, it is the process in which the bacteria use a certain sugars for their production of energy. For example, we have Neisseria gonorrhea and Neisseria meningitis. These bacteria, Neisseria meningitis and Neisseria gonorrhea, these two bacteria can be identified on the basis of their fermentation of sugar, because Neisseria uh, gonorrhea it actually ferment glucose, while Neisseria gonorrhea uh, it ferment maltose, while meningitis it ferment glucose, while E. coli it usually ferment lactose. So what is fermentation? So fermentation, it is the process in through which the bacterial cell break down the sugar molecule, and sugar molecule breakdown will lead to a glycolysis process. You remember glycolysis? So what is the end result of glycolysis? Yes, can anybody tell me?
Pyruvate.
Pyruvate, very good. So fermentation, uh, the this glycolysis, the other name of glycolysis is fermentation. We also call it fermentation. So glycolysis or fermentation, it is the process through which a bacteria utilize a sugar molecule and convert the sugar molecule or break down the sugar molecule into pyruvic acid or lactate. Okay, pyruvic acid or lactate. So fermentation is the glycolysis process, same glycolysis process to produce ATP, to generate ATP. And if you recall your biochemistry last semester, we know that the pyruvate which is produced by this glycolysis process, it enters into the Krebs cycle and produce more ATP. So that is why this Krebs cycle, the tricarboxylic acid cycle, it actually require oxygen. So all the facultative aerobic bacteria, the aerobic bacteria which ferment oxygen, we know that in the absence of oxygen, anaerobic glycolysis, it produce lactic acid, while the aerobic respiration, it produce pyruvate, and in aerobic respiration, the pyruvate enter into the Krebs cycle and produce ATP. So ATP generation by the Krebs cycle produce actually the Krebs cycle, it produce more ATPs as compared to anaerobic glycolysis. So that is why the aerobic bacteria, facultative aerobic bacteria, they grow very rapidly. The facultative aerobic bacteria, they grow very rapidly. Why? Because oxygen, because of the oxygen, because of the Krebs cycle. So this is we call the fermentation. So during fermentation, when the fermentation it occur in the bacteria, this fermentation produce pyruvate or lactate. Anaerobic bacteria produce lactate, aerobic bacteria produce pyruvate. So these pyruvate and lactate, these are the acid, so they change the pH of the media. So we can use different indicators if we want to detect whether this bacteria are aerobic or anaerobic. So we can use some detectors to detect the pH change. So by detecting the pH change, we can identify that whether this bacteria is uh, aerobic or whether this bacteria is anaerobic. So we have different biochemistry or biochemical reactions, as well as we have some chemicals which detect the pH change when we grow the bacteria in the laboratory. So next we have the iron metabolism. Remember, iron is also required for the growth of a bacteria. It is also essential. So pathogenic bacteria, we have pathogenic bacteria. When I say pathogenic, it means the bacteria which is present in our body. or the bacteria which are outside the body, the bacteria which are outside the body, they can acquire iron from their environment. But the bacteria which are present in the body, in our body, they cannot get iron very easily. So iron is required for their cytochrome and enzyme system. So that is why iron is essential for their growth. In human body, the iron is, the free iron is very low in number, a very low amount, because in our blood, in human blood, in our blood, the iron is mainly present in a binded form. So what is the name of that protein? Physiology you have studied.
Apopharatin.
Which one? Iron binding protein. No, that is a
Sir,
hemoglobin.
apopharatin.
No.
Ceruloplasmin.
Transferrin. Transferrin. Actually, transferrin, it is the reservoir of this iron. It's an iron binding protein, transferrin. So because the iron is binded, so because iron is bonded with the transferrin, so the bacteria cannot acquire the iron very easily. So for this problem, bacteria produce another iron binding compound that is known as siderophores. It is written here. Yeah, here. To overcome this problem, the bacteria, it produce the siderophore. So siderophore, it is a protein, and this protein, it capture the iron by chelating. It actually capture the iron, and then it attach to the specific receptor on the bacterial cell surface, and then it will be transported into the cell. So the bacteria, it produce a protein that is known as siderophores. This siderophore, it capture the iron from outside the cell and then attach with the receptor on the bacterial surface and then transport the iron into the bacterial cell. So that is why this iron is transported and utilized by the bacterial cell. And remember, iron is very important for the bacterial growth. Okay, so bacterial culture, should I continue? Okay, so bacteria can be now the third, you can say, section of our this lecture is the culture. First, we have discussed the bacterial growth, bacterial nutritional requirement. Now we are discussing about uh, the artificial medial growth media. Actually, this is not your theory. It's not your theory related uh, topic. This is actually your practical related topic, because we studied these all medias in our microbiology lab. So I don't know whether you will have the labs microbiology lab or not, but this is actually a lab topic. So anyhow, bacteria, uh, if we want to culture a bacteria or grow a bacteria from outside the body, like outside the body, we need to provide the nutrition to the bacteria so that the bacteria can grow. We need to provide the medium, we need to provide the nutrition, we need to provide the environment to the bacteria for the growth. So for this reason, we have different kind of media. Media means the solution or the culture or the environment or the nutrition, mixture of nutrition, which can provide nutrition to bacteria for its growth. So according to the media which we the media which we use in laboratory for bacterial growth, there are they are basal media. So this basal media, it contains simple nutrients for the growth: water, peptone, and broth. Remember, not all bacteria can be grown on a simple media or a single media. Different bacteria, they have different requirement for their growth. So that is why we require different media for different type of bacteria. So basal media or a basic media, the basic media, it is actually made up of agar. It is also known as agar media. AGAR, agar. So this agar media is a basal media which contain nutrition, water, peptone, and broth. So this agar media is available. This agar media is available in two forms: semi-solid, and in the form of broth. What is broth? Liquid. So both forms of this media is available, and we can grow bacteria on the Petri plate. The Petri plates are the plates on which we grow the bacteria. So we pour the agar and let the agar to solidify. So it's a kind of semi-solid to solid because it form a gel on a Petri plate, and we can grow the bacteria on it. Then, and the broth, it is a liquid form. Is the same agar, but it is in a liquid form. So we can grow a bacteria on it as well. Then we have enriched media. So enriched media is the same basal media, but it is the blood is added in this media. You know, E. coli bacteria cannot grow on simple media. If you want to grow E. coli, we have to use this enriched media. So those bacteria who require which require blood for their growth, they need this enriched media. So we culture the bacteria on this enriched media. Then we have differential media. So this media also have indicator. So that indicator sends the aerobic or anaerobic bacteria. So it can tell us which colony is a the bacteria of aerobes, or which colony is anaerobic bacteria. Selective media mean that if we have a sample, if we have a blood sample or any patient sample, so we don't know what type of bacteria in it. So there are a lot of bacteria may be present in a single sample. So if I only want a specific type of bacteria, so I will use some selective media. In selective media, only one type of bacteria will grow, while rest of other bacteria will die. So that is we call selective media. And anaerobic media, that media which lack oxygen. So that media, and that is actually the environment which helps the anaerobic bacteria to grow in the lab. So these are the media for artificial growth of bacteria in the lab. Okay, so it's another topic. So we will continue this topic in our next class because we only have 6 minutes left. So I need to take your attendance. So meanwhile, if you have any question, please you can ask. Any question? Okay, roll number 2. Your attendance, please.
Present.
3.
Absent, sir. Absent.
Okay. 4,
Absent, sir.
6,
Present.
8,
Present.
9,
Present.
10,
Dan dosa kedua orang
13.
Present.
Wait. Hemoglobin is a oxygen carrying protein which is present in the blood. Transferrin is a protein which bind the iron. It is also a circulatory protein. It balances the iron. Okay. It's different. 14,
Present.
17,
Present, sir.
18. Please be
Present,
quick.
sir.
19,
Present, sir.
20,
Absent, sir.
21.
Absent, sir.
20 is present.
Absent.
ये देखो ये टेंस हो
22,
Present. Present.
23,
Present.
26,
Present.
28,
रही है जल्दी आओ
Present.
33,
Present.
34,
present
37, 37,
Who? Present, sir. She was present.
38,
Present.
39, 39.
He was present, sir.
But he is not present now. 42,
Present.
43,
Present, sir.
46,
Present.
49,
On leave, sir.
50,
Yes, sir.
ठीक है
54, 54,
He was
50.
present, sir.
He was present, but he is not present now. Where he is? Okay. 55, 55.
He was present, sir.
Yeah, he was present. I saw him. 56,
present
here's 61,
Present.
64, 64. Please be attentive.
He was present, sir.
65,
Present.
66,
present
67,
Present.
69, 69,
He was present, sir.
60, 70, 75,
Present.
76,
present sir
77, 77,
He was present, sir.
present था ये भी
78,
Present.
80,
Present.
82,
Present.
83,
present sir
84,
present
92,
Present.
93,
Absent, sir.
94,
present
96,
Absent, sir.
98.
Present, sir.
Okay. Okay, class, any question?
sir